Bismillahirrahmanirrahim. At the very onset, I would like to submit my sincere obligations to Professor Nadeem for his kind words for me. I am also sincerely thankful to Professor Iqbal, who is my friend, and I am also highly obliged and indebted to this institution, which, honestly speaking, is doing the great job of educating people to the core. Now, let us, without any waste of time, come to today's topic, and that is gene therapy, the future of medicine. Since this would be a lecture in a sense that it's a discussion whereby you will not only interact at any given time, and you should have the liberty to stop me anytime by asking any query regarding the material which I am projecting to you. But also, since you all are sharing the journey of knowledge, we are therefore colleagues. So, my dear students, don't hesitate in stopping me in between and asking questions. Although the core of the forum is that you and those who are online would be given an opportunity to ask questions at the end of the session, and I'll try my level best to come up to the expectations of their queries. And it will be a matter of 10 to 20 minutes for that as well. So let us see what is gene therapy. Basically, gene therapy is a medical approach that treats or prevents by all sorts of disease. Let me add to this. All sorts of diseases. I stand in front of you with confidence to say this, that gene therapy as defined today is a medical approach that treats or prevents diseases. And by diseases, I mean all sorts of diseases, not genetic diseases, by correcting the underlying genetic problem. Now I know you are not asking me, but the question arises that all sorts of diseases are having genetic problem. My answer to the question is yes. Yes, all sorts of diseases are genetic diseases. Therefore, you can understand my topic and the importance and significance of this. Gene therapy has the potential to treat diseases that cannot be treated with conventional medicine. This is an edge. You can treat the diseases by conventional method. By now, you only think that infections, antibiotics, tumors, surgery. Now, that you are talking about past now. I am talking about the future. In future, infectious diseases, all sorts of cancers are going to be dealt with gene therapy. You may be thinking that I am exaggerating. Let me proceed with my slides. Your concepts will be changing. And I am making the basis of my news further slides. So, what is gene therapy, therefore? It is applying and transferring the nucleic acid which has gone wrong into the patient's cell, or in other words, modifying the gene. The gene which is mutated, which is abnormal, which has gone abnormal. Let me use a word here with all your elite audience. Gene which has rebelled. Fine. Now, before I go into detail, this slide I love. What a wonderful slide it is. You are thinking that I'm appreciating myself. No, just go through it. Imagine we all have been through this. When we were small, we used to play. And when we used to play, it usually happens that you may have broke the neighbor's window. It could happen. You are playing cricket, or for that matter, any game, and you have broke the window of the neighbor. Now, what your options are? And the first option we are doing in medicine, we stay silent and we think that no one will ever know it. No one will ever find our guilt. But what happens to the window? Window is not fixed. It remains broken. Although fact is that because you are silent and you have escaped, you will not be blamed. This is one way. This way we are following. Second is that you repair the window with some tape. The glass is broken, you put some tape there and run away. This is what we are doing in surgery and antibiotics in medicine. Broke window, we are just taping. Third is that you are dare enough to go to your home back and tell your mother and father, I have broken the window of my neighbor. Please help me. They go and bring another window and permanent solution. Neighbor is happy, you are happy. I dare saying here, the neighbor is your nature. The creator who has created all this with good genes and the bad genes. But we have been keeping ourselves silent. Imagine silence at times is neither silver nor gold. So repairing is one phenomena, putting up a tape is another way, and fixing a new window is the permanent solution. Now, same is true. Stay silent is ignore the genetic disorder that is in you, that is in your patient. And you never fix it. Second is you try to treat whenever there are symptoms and signs of the disease by drugs, by surgery, and by other resources and approaches. But the third one is putting a new window, and that is a normal functioning copy of the gene. So now you understand that we are talking about future. Let's start from the beginning, because once I say gene, we should know what is gene. Just for one, as I said, it's not a lecture, it's a discussion. And I always love to interact with the back benches. I do not know why. Because I know this, that the brilliant students, they always occupy the front seats. So the back benches, I would like to just ask, what is a cell? That is it. So my concept is wrong. Back benches are also brilliant here. Wow. I know the institution is great. So the biological unit of life is cell. And I'm sorry, I am going in details of the basics so that I can build up my what I'm going to tell you in near future. Basic component. Can you imagine a cell without a nucleus? They all say yes. And if they all say yes, there will be a difference of opinion among yourselves. A cell without a nucleus? So let me ask, what is a nucleus? What is a nucleus? So the answer is half the way long, half right and half wrong. See, when you define life, you said basic unit of life. Life is in plants, life is in animals, and life is in between. We still have no stand to tell you that the fungi, the algae, the basic unicellular lives, though they are the basic units, but they are alive and you have no demarcation. No, let me, because since I am a bit difficult, let me make it easy for you. Basic unit of life is cell. What is life? I tell you, now I'll face a long, long silence, whether back benches or front benches. But how can you imagine when you say that plant is alive, animals are alive, unicellular or multicellular, when it comes to virus, yes, life. Oh no, not life. What is it? So the difference is nucleus. In the nucleus, there is nucleolus. This you will agree that some cells have nucleus, some cells have nucleolus, some have not. Let me come to my next slide. So, what I am saying is definition of life is still not defined, because all those definitions that it can move, it can adapt, it can regrow. When it comes to certain species of viruses, you are at a stunt, and some say alive, some say not. So this dilemma I am going to solve today. Two main categories. Now, if we categorize cells into these two types, then the difference is in nucleus. Can you imagine a nucleus without a DNA? So this brings me to tell you what is DNA. Whether there is nucleus or not, this is agreed that there is a DNA. And in the virus, there is a DNA. And now we are coming across with certain viruses which are only messengers, that is RNA. That is why science has to revolutionize all the definitions. And because I am talking to PhD people, I am opening up the chapters for you. Think, go home and redefine things. This is how we progress. Today we are challenging Mendel's law. Why? Because over a period we have come across with our research which are against them. Yesterday the beautiful method of identification was your fingerprint. Now fingerprints are not available in 60 years old man. You are saying epidermis, the dermis is gone even. So the identification is not only with fingerprints, then comes the iris impressions, and with cataracts that is also gone. So now this is DNA, the identification mark of individualities, of personalities, of life. Prokaryotes lacks a defined nucleus. We have talked about it. Defined nucleus means nuclear map. Simple enough. So I would now say you cannot imagine a life without DNA. I have come to my definition. I have come to my point. The other type has got a membrane, a surrounding membrane. But the fact of the matter is, both do contain the structural unit of nucleus, that is the DNA. So let me come to it, that what is DNA? You may be defining cell has got a nucleus. In the nucleus there is a DNA. This happened our teaching. Now things have changed. I am jumping to it. Quantum jump has been given to our understanding now. DNA is nothing but a gene. You would like to add for me today that no, it's the code. I'm coming to it in a minute. But in simple words, I define gene as the DNA. That may come to it. And till now, any disagreement? I am giving everything logically, right? There is no controversy. Great. So it means that all of you are sleeping. No. It also means that all of you agree to me. The genetic information is stored in DNA. Now let us see what this DNA is. It is in its term defined. D, oxy, ribo, nucleic acid. We can put the same thing in the structure, right? So all of you can now draw the DNA. So can you draw all the genes? So why don't you make controversies at the stages when I am speaking to? But I know that when I speak on it, you agree to that. What I mean is that DNA is nothing but a chain of those molecules, either of the sugar or of the amino acid, the units of life. What is life? Same like what is food? Carbohydrates, proteins, and fats. You have to categorize all those foods into these three. Similarly, all the proteins are built up of amino acids. And the DNA is going to code the protein synthesis in its own way. Mind you, I have finished the lecture. If you agree to this, then rest of the slides you will understand like so simple. Now, when I say this, I mean to say that we are in the era of molecular basis of diseases. Understand that. Our forefathers are not able to do this because they don't know how to see these things. Their tools were not able. So when Mendel speaks of inheritance, he had no knowledge of gene. Gene in the sense of molecular structure. And that is the reason that now every third scientist of genetic therapy is saying against it. You have coined the term. Neo says that mitosis has gone back into the past because of our understanding of molecular basis. So when I say gene therapy is the future of medicine, I mean to say molecular treatment of diseases. Fine enough? Any disagreement? Anybody who has some controversy? No. Okay. DNA contains all the information. And by the way, when we say all information, all coding, this blah blah is in DNA, how is it when the fingerprints were having patterns identical to every individual? Similarly, DNA is identical to every individual. By what? By the sequence of the molecular structure in which the DNA is formed. And that is the gene. The sequence code of DNA of an individual is the dictation of the building up of the protein synthesis throughout the life. So, a gene will indicate that this gentleman will be having diabetes mellitus in the year 40. And you are meeting him at the age of 20. There is a broken window and you keep silent. When the 40 will come, he will get diabetes. This is how we are keeping silent. Now is the era when you say no before it happens. I will refix that code, that information which is in that gene, which is going to make him diabetic, will replace. Agreed? So we are talking about DNA. What is DNA needed for? I think all of you can answer now. DNA is needed for information of gene expression in the future of the person. And I am talking you to give you information of the therapy of the future. So would you like to keep silent or would you like to refix the window? Choice is yours. And what is RNA? Okay, it's really difficult for me to face very silent audience. I don't know why, but it's with me. What is the difference between DNA and RNA? Any brilliant student, just he will be recorded, introduce himself or herself and tell me the difference. My question is difference between DNA and RNA. Please, can you just introduce and answer? Fine. IT, you have explained my diagram. See, that is what I am showing. So it was right in front of you. Now, imagine the diversity in the unity of DNA. DNA is composed of four bases. These four amino acids with a sugar base, but this structure is speaking high of life. No life without DNA. We are not defining life with plant, animal, and other species or structures. We know more. People are even now outspokenly saying the crystal of the diamond. They are debating of their life. Imagine. So we have to redefine everything. The sequence of this amino acid with the base and sugar is going to make the sequence of the gene. So simple is this. And when I say sequence, I mean code. The genetic code describes the sequence of the gene which dictates to convert protein synthesis. So simple. And when we say code, the unit of it is codon. By the way, when I saying this, let me tell you, there are trillions of cells in your body, more than that. And every cell we are talking molecular basis. So imagine a cell itself has more than 13,000 genes because of the sequence, and that is going to affect. And we pick it, restructure it, and put it back. And already when I tell you the history, things are being done. So because my topic is gene, let me take some more time on that. When we say gene therapy, it means genetic engineering. Genetic information is encoded in the basic sequence or structural sequence of the DNA. A gene will dictate the DNA sequence either by coding it or uncoding it. And from that point of view, we are taking the whole subject for its therapy for the diseases of human beings. And my diagram is so clear. I'm talking about trillions of cells of your body, and I have taken one cell to give you the sequence. Cell in the nucleus, the DNA. In the DNA, the chromosome. In the chromosome, the structured DNA getting its sequence, and that last side is I'm interested. From there we pick and we change the sequence. Let me more elaborate in my next slide. For that, I would like to introduce to you two terms. And for that, I will take help of one of the most famous, especially in ladies, a famous Urdu proverb. Two important terms, phenotype and genotype. The phenotype is the basis of that old Urdu proverb. That is, कटोरे पे कटोरा, बेटा बाप से भी गोरा. Can you answer me for what this is? Now it is much available. Nariyal, coconut. But how could this is a proverb? Forget. I want you to go back to my lecture. The point is, those people they were knowing that. Those old ladies they were knowing the recessive and dominance genetic hierarchy by saying kotore pe kotora, beta baap se bhi gora. Have you seen a beta baap se gora? Imagine if you don't know gene, that is the dominance, and this is nothing but phenotype. Phenotype means things which you can see. गोरा है, खूबसूरत है, तिल यहाँ बाप का भी है, बेटे का भी है, and so on and so forth. The outlook, the impression, the structural outlook is phenotype. And our knowledge stayed there for hundreds of years. Our genetic was only that much. माँ की शक्ल पे गया है पहले दिन ही बुरी औरतें आके क्लेम करते 10 दिन बाद देखें तो वो बाप की शक्ल पे imagine. But from there, the basis is there. I salute those old women. They have given us the foundations of genetic hierarchy. But the genotype is marvelous, unhidden inheritance. And that is my point. बाप को डायबिटीज है बेटे को तो होगी रिश्ता ही नहीं करो imagine even they were knowing that so those hidden hierarchy is genotype my concern would be in future of only genotype since we are talking about human therapies of future this is basics 46 chromosomes 2 sex chromosomes making it out 44 autosomes and 2 sex chromosomes. X and Y, you all know, XX female and XY male. This is basics. You know all this. But I am now challenging the Mendels in the way that you will appreciate in future of my slides. That we have two copies of each gene, one from the mother and one from the father genotype. Dominant character, one allele needed to cause the phenotype. यानी ये भी तो हो सकता है ना बेटा बाप से गोरा नहीं बिल्कुल ही काला है so the concept is so clear the problem in gene therapy is this that there is a big debate now if we go for the germline inheritance I'm coming to it in the future recessive character both alleles need to cause the phenotype so this explains the features of the father in phenotype will be either recessive or dominant will give you the idea. On this basis, we are going to select our patient's therapies. Hierarchy of genetics remains the same, but the future of medicine regarding the therapy would be genotype. And debate starts when we come to the germline cell genetic engineering. This all is known to you, and I have been across the world, and I have really noticed this. Definitely, I have put this slide because the population inheritance is not environmental. A whole area of the geography of a region, you will find similarity in genetics as well. People used to think because of the environment. Now we come to know, no, this is population inheritance. The South Indians of Asia will be identical. All Pakistanis, whatever the difference, you go all around the world, you will identify them, though they are not in the environment. And this is really something stimulating. Coming to the diseases, as of now, I mean, we are talking of the past for the last 20, 25 years. The hemophilia, muscular dystrophy, and cancers, and to some extent hyperlipidemias, they have been the subject of research. And we have been successful in treating these diseases genetically. But this is the smallest spectrum for the last 25 years. The quantum jump in the last 5 years is that it has opened a new horizon. A new horizon. It has actually revolutionized the whole medical era. And this is what I have told you. Our understanding of molecular basis has really made a big change. A change which will change the world. Advances in molecular biology have triggered an unprecedented expansion of knowledge. And you know, knowledge is now global. Whatever is being done in America is next day to us, although we are sleeping. So this is one very alarming. That if we lag behind, we lag behind. We should change our courses of syllabi as well. Because many things are going to be redefined. Gene therapy is a medical approach that treats or prevents diseases by correcting the underlying genetic problem. No. So it means only genetic problems are to be dealt. I come forward and I tell you all diseases have some genetic problem. I've given you the example of that broken window. I come in future and I would like to tell you that we are going to treat diseases before they happen. Let me tell you, I'm not talking about death. And this brings to my mind the holy saying of the Holy Prophet that all diseases are curable. The earliest method of gene therapy, often called as gene transfer or gene addition, was developed to introduce a gene into the cell to help fight the disease. Now, this you will not understand till we come that how do we do it. So up till now, it's all theoretical for you. Now, let me come to give you the practical aspect of gene therapy. So this definitely brings me to tell you how does gene therapy work. It works by altering the genetic code of the critical protein mutant gene. As you know, proteins are the workhorses of the cell and the structural basis of the body tissue. Fixing or compensating for disease causing genetic changes may recover the role of these important proteins and allow the body to function as expected. Now, gene therapy actually can compensate the genetic alteration by recurring and refining it. So simple as the gene transfer therapy introduce new genetic material into the cell. Now the point is that in the last decade, we were not able to approach the cell at the molecular base. Now we have got the weapon, the one which can go into the cell. We put the gene into it, and who can go into the cell? If a mosquito bite can get into your blood, so virus can go into, so vector is virus, one way. But this also we are talking about the last 30 years. Now we have other tools as well. Exactly. Now things are clear. If you can approach a disease cell and you can take out the gene and you can put a new gene, you need a vector. The problem with the virus was that this virus changes, mutation. If you overcome that, you're right there, but you are talking of the future. So alternatively, the therapy can introduce a different gene that provides instruction for the protein that helps the cell function normally. And I'm speaking of cell of trillions of cells of the body. Identically, one cell I have taken to talk to you. The principles of gene therapy are number one, gene modification, that is replacement treatment, replacing a natural gene with a non-natural gene. Second is modifier gene therapy, restoring the natural function to a defective gene through selective reverse mutation. So if you are capable of directing a virus, that is a vector, you can do anything with it. So we have modified all sorts of mutation for this viral vector. This is one aspect. This diagram you just concentrate. When we say a cell would be either in the body or also you can take the cell and bring it to your laboratory. So vivo and extra and in is the concepts, and the diagram is showing you even a syringe. I will stay on it for a minute so you understand all this. But this is so beautifully explained. Either transfer of a gene into a patent cell outside the body or transfer of a gene directly to cell inside the body. Vectors. So we have talked about one. So we will come to no viral also. Viral, you know all this, that retroviruses, adenoviruses, adeno-associated viruses are some commonly used viral factors, whereas Some less commonly used viral factors are derived from the herpes simplex. But when we say virus, especially post corona, we are all shivered. We should come out of all those fears now. No viral vector. It involves the chemical and physical methods such as direct injection of naked plasmid DNA by bombarding and receptor mediated and gene transfer through ribosomes. All this is not anymore future. I am talking of the recent advances. Types of gene therapy here, actually the problem is the second one, that is germline gene therapy, which is to some people non-ethical. Let me read out the whole slide for you. Somatic gene therapy, the somatic cells of a patient are targeted for foreign gene transfer. In this case, the effects caused by the foreign gene is restricted to the individual patient and not inherited by the patient's offspring or later generation. Now, this has been known to us for the last 50 years when we are combining tiger with lion and all those things we halagula we are doing. We come to have a new animal, but that animal cannot produce. So from there the lesson is. But now coming to the germline gene therapy, which is debatable in the ethics, I will come into a minute. Let me redefine it. The functional genes which are to be integrated into the genomes are inserted in the germ cells, that is the sperm and the ova. Targeting of the germ cells makes the therapy heritable. So it goes into the offsprings. At this point, the world has been and still is having controversies. Some people oppose that and some not. The others ways and means and methods of gene therapy are I enumerated here. So other than virus factors, the other one are all this. The last one is more applicable. If you can just inactivate that you have identified harmful gene, then the disease is over. The editing technique is a new recent development. You can edit the genome. And now I come to know to the conclusion by saying that you have to just change the sequence of the pattern of the amino acids in the gene at the molecular level. The last terminology is known to us for the last 20 years. I have just given a history just to know that how rapidly, progressively, and steadily we have been doing gene therapy, and for the last 5 years, what a quantum jump! What a quantum jump! It started somewhere in 1990 in September by this D. Salva young girl with a rare genetic for immune deficiency who was the first recipient of human gene therapy. Her own white blood cells were genetically modified in the laboratory by inserting a normal copy of the faulty gene before being reinfused back into her body. This is the start of 1990. And as you know, the start is always having some challenges. So the effect was transient. But then, after that, approximately 1700 clinical trials on patients have been performed, and I would say successfully, with various techniques and genes for numerous diseases. Some are I have jotted here. Of course, leukemia and Parkinsonism, quite good success, quite good. Further research, cancer, including all these, but to summarize, all sorts of cancers, neurological disorders, inherited diseases. I'm coming to this thing. When I say infectious disease, so it means gone would be the era of antibiotics. And I know that you'll be surprised. I have written cardiac diseases as well. Can you imagine we are treating tuberculosis, malaria, HIV with genetic engineering? You are familiar with genetic engineering in the inherited? No. The application of gene therapy technique is by no means limited to inherited disorder. In fact, over half of all current gene therapy trials are for the treatment of non-inherited cancers. I want to brainwash you from the fact that when we say gene, we come to the conclusion of inheritance. But yes, there are challenges. Who is going to be afraid of the challenges? My point is, we have to face the challenges and overcome the challenges by constant research work, immune response, targeting and delivering issues, long term effects, and safety. Safety is our concern. And of course, the ethical concerns, which is only for germline editing because it is transferable and inheritable. This slide, I would stay another 5 minutes. Every point we will discuss. Are you familiar with the word suicide genes? The genes that go in and suicide means change them. They get their fate. But these people of the tendency is for healing. So the tumor therapy would be the insertion of the suicide gene into the tumor to promote tumor cell death. As you know, the antibiotics are killing the bacteria and the fungi which have invaded and made the body infection. Another approach utilizes the immune system to kill tumor cells by altering tumors so they bear surface markers that evoke a strong immune response. And this is being done. Infectious diseases are another potential target. So this would mean goodbye to antibiotic era. When we say cardiovascular, I was surprisingly noting that nobody objected, but I have explained now. Many of the diseases of cardiovascular origins are because of the thickening of the artery. And we are going to treat them by gene therapy. All of you have gone through the slide? Good. Now the recent advances, and still things are going on. Because every day we are reviewing all these recent advancements as well. So the ongoing clinical trials promises the future prospects. Now, do you agree my title? The gene therapy is the future of medicine. You all agree? And if you agree, it means I am a successful presenter. So the ethics aspect, I am just leaving because it's debatable, controversial, but sooner or later it will be solved. Conclusion. Gene therapy is a novel, not only novel, it's a novel approach to treat, cure, and even prevent diseases by changing the expression of a person's gene. Typically, gene therapy involves using a vector such as a virus to deliver a therapeutic gene to the appropriate target cell, a cell which is diseased. Gene therapy is still in its infancy. But I tell you, this is going to jump from infancy into adulthood very soon. Gene therapy has originally started with treatment of monogenic disorder, but majority of gene therapy trials now all over the world are challenging diseases of cancer, infection, and cardiovascular origin. Although it has been controversial ethically, but sooner or later, all the ethical problems in the world regionally will be solved. And controversy is always a doorstep to success. Before I thank you, I would like first to give chance to those who are online to ask questions if any, and then I would allow the audience to ask questions, and I'll try my level best to answer satisfactorily to the queries. So the forum is now open for the online listeners. Is, is it available for the people? Just ask somebody who is outside, who is managing this. Like, can they ask questions?
Can I ask the question? Assalamu alaikum wa rahmatullahi wa barakatuh.
Yes, you can answer. You can ask questions.
My question is, what are the what you call risk factor associated with the gene therapy?
Kindly repeat the questions.
My question is that what are the risks associated with the gene therapy? Gene therapy.
A very good question. I am thankful to you. The question I would like to repeat is that what are the risk factors in gene therapy? At the moment, there are so many risk factors, and I have put these risk factors in the term of challenges. And one by one, we will overcome these challenges. Say, for example, one of the risk factors is that in the germline therapy, we treat a person with mutating the germ cell, that is the combination of zygote, the egg and the sperm. But that in the future would also be inherited. What a great risk factor it is. And that is why people are against it. In fact, many countries have put a ban on germline genetic engineering. Any other question? So we'll allow our audience now. Any question, please. Number one is that the last portion of your question, I will not agree that we don't have the treatment for viral. We do have treatment for all viruses diseases. In fact, topical and non-topical both. And number two is that is the whole point. That viruses, they mutate. And we have to overcome that mutation. In fact, what we are doing is, what you people are doing here in your research center, you are dealing with drugs which were targeted for one disease, and then you saw that drug side effect, and that side effect you pick and you repurpose the drug. That is what you are doing, repurposing of the drug. Similarly, the repurposing of the mutation of the virus is genetic engineering. Instead of the virus deceiving you, we are deceiving the virus. Any other questions, sir? If no questions, it means that the presentation is fully understood or fully not understood. I am again thankful to Professor Nadeem, and the forum is back to him. Thank you very much, sir. Thank you very much.
थैंक यू डॉक्टर साहब, बहुत बहुत शुक्रिया आपने इतना करंट टॉपिक्स पे डिस्कस करना, और सबसे इम्पोर्टेंट चीज़ है कि आपने इतनी दूर से आते हैं हमारे लिए